Good evening, everyone. Welcome to NyBerMan tutorial on bioinformatics literature search and database tutorials. So today we'll be seeing about tools and databases and literature materials which are available, where to find literature, how to curate them, how to pinpoint to the research which we want to conduct, and what are the databases available. But we'll cover not all of them because it's like very time-consuming one. So today's agenda will be I'll just speak about a little bit about the basics of bioinformatics, not much into the details, and then we'll proceed into literature databases and then literature tools or servers which are available actually. And then some of the tools I'll show you where you can collectively see the resources basically. So let's start with bioinformatics. So basically, there are multiple definitions to bioinformatics as well as computational biology. So in basically what we are trying to do is we are trying to analyze biological data using computational tools. So these biological data can be either sequences or structures. Sequences can be basic raw sequences from your sequencing machine or a process sequence from other resources like from secondary databases also we can do. And structures basically refers to protein structures or macromolecular structures basically. To understand the biology between these sequences, structures, and these interactions, we use concepts from various other domains like mathematics or statistics, and the force fields and the functions between them are defined based on the laws from chemistry and physics. So these are the like foundations for bioinformatics. So why do we study biology? Is to basically understand how things work, to understand any mechanism. Interestingly, we are mostly looking at understanding diseases. So when we come to diseases, we mostly look into identifying particular genes or proteins as targets. So there are other things from like except proteins and genes like metabolome, like non-coding RNAs, all those things are also available. So these proteins can be classified into targets, and the compounds or the metabolomes or metabolites or the chemical entities are defined as compounds. So these compounds can be further developed into drugs. So this process is mostly into the computer-aided drug design. So SPCAD is like structure-based drug discovery approach. So structure-based computer-aided drug discovery or drug design. So basically, when you study omes, like collectively genes or proteins or transcripts, anything, so that is termed as omics. So this omics field is basically based on the sequences, and the CAD field is basically from the structure point of view. So these are the various basic things which we do in bioinformatics. So to start any study, we need to understand what has been done so far. So for that, we use literature resources and tools. So there are open resources which are available so that we can use for our research purpose. So this is one of the image which I took from the internet. So which collectively tell about the differences in biochemistry, chemoinformatics, and bioinformatics. So here you can see bioinformatics lies in the intersection of biology and informatics. So we are trying to understand the information to solve biological problems basically. So to begin with literature search, we will start with two concepts. One is journal and publisher. So publisher is a bigger or higher hierarchy in the line. So each publisher can have multiple journals listed under them. Say for example, Springer. Under Springer, we have different journals like Virus, Gene, Nature, and etc. So there are many journals within a given publisher. So these journals publish articles. So these articles are of various types. So various types in the sense like original research article, review article, editorial. So each of these articles have their own style of style and structures basically. So to publish any article, there are two models. One is open access model where the author has to pay to the publisher to get the paper read by the reader for free. And the other one is subscription model where the reader has to pay to the journal to read the article. So these are the types of articles like we will see further in any journal. I can show you an example. And these are the two modes of publication. So if we see generally to search anything, there are various resources of literature available in the internet, which are deposited in databases. So the most common one used for biological purpose or medical purpose is PubMed. And the second one is PMC. PMC is PubMed Central. So the basic difference between PubMed and PubMed Central are PubMed Central articles are open access articles. So these articles, whatever available here, is for free to everyone. So anybody can read. The full text of the article will be available. And in case of PubMed, we get articles. It's kind of mixed. You can get full text articles also the subscription based articles also available. And if your institution or university has access or paid subscription to journals, then those articles become accessible to you also. So other sites are like to search any literature. Other sites are like Google Scholar. So Web of Science is mostly used to evaluate a journal's impact or how far the journals have published. To know the details about the journal, you can use Web of Science. Also, like currently UGC has also list of journals. So similarly, the impact factor are calculated by Clarivate institution, which is under the Web of Science team. So other resources like ScienTerrect and Scopus. Scopus is a subscription based model. If your institution has only, you can access them. So now I'll switch to browser. So if you can let me know if it is working or not, I'll just switch. Okay. Can you see my browser? Okay, somebody has sent a chat. Okay. So basically, the first thing what we do is like generally what we do is like if we are say for example, I'm interested in understanding cancer. So I want to search for cancer literature. What I do is cancer. I'll just cancer paper. I'll search or something related. If I know that some target, say P10 is my target, something I'll just search. So mostly I get results from NCBI articles, which is PMC. In general, you can get articles like the resources which I listed, like Science Direct, all those stuffs which we get like based on the just simple Google search. But to get a comprehensive resource, like if you want to know from this year to this year what are the article published, then we go for such literature databases. So one of them is PubMed basically. So here in this one PubMed, you can use the same keyword search basically. So instead of paper, I'll just use the keyword cancer. Maybe I want to see drug discovery. And since we are doing bioinformatics, so you can use the keyword bioinformatics for this one. So I get 2,731 results for these keywords which I used basically. So this will create a keyword combination inside the database and which lists me the literature or the articles published. So since I have clicked the filter, so this left side you can see the filters. So filters are nothing that which can sort out articles which are based on your query basically. So this is listing from 2001 to 2022 basically. So if we move this bar, you can limit the articles from 2009 to 2022. So you can see the results have changed. So, and then I have clicked free full text. So if I remove that, you can see the article number has increased to 4,078 results. So in here, you can see the type of article is review. So here is review. So article type also can be filtered from here. So it can be book or document. It can be clinical trial, meta-analysis. It can be a review, or it can be research paper. So instead of this bar filter, we can also use the date filter based on the radio buttons given. Also, you can customize them. So in this case, I'll give free full text. So whatever article I am getting are directed from either PMC or it is a free article. So I'll just open this one, cancer cell. So it just opened the site, and then this is a review article basically. We can see the article, abstract, keywords, everything. And since I mentioned cancer drug discovery, so it is showing me this result. So to get the full text, I have to go to the original journal site. So if I click the journal site, it will take me to the original paper with the DOI. This is digital object identifier. And this article is published in May 20, and you can get the full text of it. So if I click PDF, or you can read the, oops, sorry. You can read the article also generally. You can read the article here itself directly. And if you want to save the article, you can click the PDF version and then you get the full text of it. So this is an easy way to get an article. And if I want to limit something in the search, so I can go, like say, I can use something called as advanced search. So here you can list. So if you see, this is my last query, like what are the filters I applied. So it is saving my history basically for this session. Like first I searched with this full text as a filter. Next I changed my date range, like which is from year 2009 to 2022. So this is my last search. So it shows me how much has the result the change and what was the time it took to, what was the time in that database basically. So here I can filter based on author and other factors. So here instead of all fields, you can select. If you know any author name, you can select this and give his or her name to filter the articles basically. And it will appear as a query here. So let's take something like text word. Okay. Yeah, sure. You can ask any questions if you have any doubts. Please feel free to interrupt me. So let me go and then select some text word. Okay, let's give title. In title, I want something like, say, for example, I'm doing an COVID, let's assume. So you can add here. So in this add also, there are multiple options with and, or, and not. So and is basically both should be present. Or is like either one of them should be present. Not is like this word should be present, but not the other word. Like for example, I'll give and let's assume cancer. Okay, COVID and cancer in the title actually. So if I add, you can see how the query has turned out. So it is like COVID in the title and cancer in the title. So if you can just simply hit search here directly. Otherwise, you can build up the query more detailed if you have any specific articles to be searched, or if you have say any compounds which you are looking for, then that also you can search. So if I hit search, it will list me what are the articles in title which has COVID-19 and cancer. Since I have pre-applied this full text filter, it is showing me only those, because we are mostly interested in reading articles which are freely available for open community. So that's why we are using this option. Otherwise, you can also get articles, but currently, if your institution is not subscribed to those, you can't get those things. So you can read free articles from here. So whatever we gave the title, so it should have cancer and COVID in the title. So it is showing me the list of articles. So if I want to sort them based on date, this is based on the best match which we gave the query. So you can see the query is here. Oh, sorry. This is the query which we typed in the advanced box. You can sort here. So based on best match or most recent, if I click, it will sort basically based on the recent work. So this is July 2022. So this is the latest work, and which is a free text available. So this way you can build multiple queries if you have nested queries to be searched, like say different disease or different terminologies. So if I choose alt fields, it will basically search on different fields of a journal or an article basically. So instead of, let me go to Elsevier and then show you something. So this is Elsevier. Elsevier is one of the publisher, and they have articles basically. So as I said, the higher hierarchy is journal. So sorry, publisher. Elsevier is a publisher, And these are the journals which are available basically, like additive behavior report, additive manufacturing letters. Like there are a lot of things, but you can search the journal also here. Let's use the keyword bioinformatics. Or you can sort by subject also basically. So these are the keywords which are accepted or which are in these channels. Let me open one, maybe cell genomics. Okay. So this is open access channel basically, which comes under Elsevier only. Here you can find the recently published articles and what are the online known articles, everything. Also, you can get the details about like what it contains about the journal. If you go, you can understand what this journal publishes, what area of research they cover, and in the aim and scope of this one, you can understand basically what type of work they accept. So computational genomics, functional genomics. So these types of work are accepted in this channel, and you have to pay for it. So that's why it's called as gold open access channel. So similarly, you can find a lot of other journals under Elsevier and also in other publishers. Say for example, other publisher is like Willy. Willy is also another publisher which has a lot of journals. So one can search. So instead of going to each publisher, each journal and searching, so one can use PubMed for their search query. So one important thing is each journal have something called as index. So based on these index only, these journals are listed in PubMed or any other resources. So let me show you something. Like there is a journal called International Journal of Macromolecules. This is an Elsevier journal. So let me go here. Oh, sorry. So here, this will tell you what is the aim and scope of the channel, like what type of work they publish, and what type of papers they accept, and basically what are the requirements to submit into this channel, and abstracting. So here, if I go, it will show me where are the abstracting and indexing are listed. So if I search this channel, it should appear on PubMed because basically this is also listed in PubMed. So this is the indexing. So whichever journals are indexed in these databases, it will be showing the recently published articles from these journals. So in this case, PubMed is one of them. So if I search for this journal, the articles published in this channel should appear in PubMed. So just go, I don't need advanced query. So just go back, but I can use the keyword, sorry, I can use also another way, I'll show you. So you can see International Journal of Biological Macromolecules. So whatever published in this journal, we are getting. So other way, instead of searching this, going to advanced, here you can select journal basically, and then paste it, and it will build the query. So the to search app, you have to use the keyword journal, and that way you can search. This will list me the same thing which we saw basically, the articles published in this particular channel. So similarly, you can create multiple queries for your literature search and then read the articles to find what is missing, and then you can learn about it. So one other thing you can, if you are interested in getting periodic alerts for your research interest, then you can create something called as curating. For that, you need to be logged into this one. So this is PubMed. So if you have any questions, anything related to this, you can ask now. Otherwise, I'll just move to the next resource, which is PMC. Any questions? You can type in chat also if you have. Okay, I'll just move them. So the next one is PMC. So PMC is also under NCBI only. So this is also under NCBI. So let me show you from NCBI. So if I go to NyBerMan, here under the all databases, I can see here PubMed. So if I search anything related to any disease, say virus, cancer, I just typing the keyword. So it will go back to PubMed and then it will list me the search results basically. Similarly, PMC is also under NCBA. So here also we can do the same type of search, cancer, virus. Okay. I'm just typing keyword. It depends on what is your research area. You can type with any keyword or any requirement. Maybe I'll use bioinformatics, or you can use in silico computational. It depends on whatever keyword you want to use. So now you can see it is showing like PMC full text search results. So whatever available is showing a PDF here. So it can be directly accessed basically. So here, whatever article you search, the left side is morely like similar to the PubMed, which we saw, like publication, you can filter all those stuffs. And then this is based on the funding agency which funds for your research. Most of the funding agency pay you to make your article publicly available. So let's assume like check one. Maybe I'll go and sort it. One second. Display setting. So since I'm seeing 20 articles, I'll maybe make it 50 article and then sort by publication date. So apply. So now you can see it is published, I think 5th August. So today is like 7th August. So if I open this one, it will give me a full text directly there itself. So you can read the article directly here. Since I think I searched for COVID, it is showing me SARS-CoV-2. And if you want to download this article, you can download from using this PDF. It will download the whole text of the article. So this is, yeah, somebody wants to say something? Okay. So this way you can download articles from this PMC. So PMC also gives you access to full text, which is available from any of the journals, which are indexed in PMC basically. So this is one way of searching. Here also you have the same advanced list, advanced search queries, which you can build. Only the interface looks a little different, but the things are almost the same. So if you know the DOI, you can give. If you have any NCBI like interest date which you have known, then you can give. These are the, like say organism I want to use, select only in human. Say when you search, generally it lists you everything, mouse, everything. So say if you see the query builder is like very intuitive here. If I type human, it just builds their human. So next field I can add, say in abstract, I want something you can give like drug targets, and if you know the author name, you can give the author name, but just in text, let's assume I am giving some TP53 as a keyword, and not I can give not is also another keyword, and not I don't want this in any field. Let's assume I don't want cancer, I want in some other thing. Mostly it should work. Let's see. So since here we have no data available, like since I built the query in reverse, so that's why we are not getting anything. If I built in a different way, let's go back. Let's go here and then select all field as cancer. And then maybe abstract drug target. And I guess instead of human, I should have given Homo sapiens. Let's try that one. Homo sapiens. Maybe what was the other one which we gave? I don't exactly remember, but let's see. Maybe not I give, yeah. Not cancer. Okay, one more I missed. I think I gave TP53. I'm not in cancer. So still nothing we are getting in the full text articles. Maybe if I remove not cancer, you may get a lot of query. Let's see. Yeah, because we know that TP53 is mostly involved in cancer. So I gave like not. So I expect that it should not be present. Maybe it is listed in any other disease. Since it is very much involved in cancer, so we are getting the results for cancer basically. So if you open this one, in this one they have done in human particularly, and also you should find Homo sapiens, and you should find TP53. I think it should, yeah, TP53. So these are the things which we expected to be there, and then cancer. So this is a cancer data study, so it is available. So it is like showing whatever query which we built. So based on that, you will get the results. Similarly, you can build nested queries to constrain your search actually. So this is how you can build queries to retrieve articles for your literature search, and then from that you can like find out what are the gaps in your research area, and to further what you can develop, or you can adopt some methodology which is already published. So bioinformatics research. So bioinformatics, if I generally search, it will give me list of articles which uses bioinformatics in their data analysis basically. So since I have, I think, sorted, if you can click also summary, it will give you summary of text. You can download it. So this is the summary of the articles like which is listed there. So in silico instead of bioinformatics, and then RNA-seq analysis. So it is part of bioinformatics. So these are the things which you get. This is just for your reference. You can get a summary and do it, but otherwise you can select also. Select particularly which ones you are interested. Similar way, you can send them to a file or email it. So what you want to summarize based on which format. So Medline is a different format. XML is used for web queries. So if you want, you can get XML files also. Otherwise, you can get the PubMed central ID also. So each article has an ID. You can, if you notice here, so PMC ID is listed under each of the articles. So this is to a unique ID which is given for an article. Say, if you know any article, you can directly search. Let's copy this one. And then if I just, even you don't have to be in PMC also. If you just search in NCBI interest, you will get the article. So if I just go to NCBI and then search under all database. So you get PubMed Central 1. So if you click here, it will go to the same article which we saw. So if you have the article ID, it is easy to access. Same way, if you know the DUI, this is a DUI which is unique for each of the article. If I just search it DUI also, you will get the same article. So I think it should list me, but I think it is under newly published. So I don't know it is available or not. EPUB I had a print, but it should appear if I search based on that. Maybe the DUI sometimes will take some time to load. Maybe I'll just go to the original article and see. Yeah, let's see. Yeah, now it is redirecting. So I think I missed the HTTP part, so that's why. So this is the original article which we saw there. So these are the ways to search, and then like to limit your search or to query your search, and then like maybe make filters in your search. So the recent activities can be tracked here. And if you have any questions, you can ask now for this part of this one session. Any questions? Okay, I assume there are no questions, so I'll just switch to the next one. So next one is Google Scholar. So Google Scholar is also like similar to PMC or PubMed Central, but one thing is that Google Scholar accounts for everything. Most of the journals, most of the things are available in Google Scholar. Also, you can also sorry. So you can also search things or search articles or search author names, and basically you can find literature which are listed under Google Scholar index basically. So these articles are my recommended articles. So it's based on my searches and my research area it is suggesting. If you want, you can search also basically by typing whatever keyword you want bioinformatics. Let's assume bioinformatics analysis tool. So these are the things which are suggestions from Google Scholar. So bioinformatics analysis of gene expression. If I search for this one, so I get articles. So directly I get PDF link or a HTML link to the original journal site. So this is basically from European Review, and this is from Science Direct. So here also you can sort by date, and then you can filter by year also, and type of article. You can include patterns, you can include citations. So since Google Patterns is another server for searching patterns, so here they are listing out the patterns also. So here also you can like search based on your interest of range year, like from I want from 2010 to 2022, then you can do that also. If you search, it will show me those articles only. So one advantage here is like it will show you like what are the articles which are citing it. Say one article is cited by other article, so you can get the cited article also here. Also, what are the versions available? Like if it is available online, and then it was corrected version available. So these are tracked, and then it will be available under Google Scholar. But again, you have to look up these articles are full text available or not. So that could be one trick where like you don't know these articles are available for full text or not. So this journal is open access. So this article will be available freely for you to read. So otherwise, most of the things you may not get. So let's go to this value one. I think this is not available for us to read. Either institution or somebody should pay for it. See, institution access is not there. So you have to pay either the listed amount to read this article. Otherwise, there are other ways which you can try, but this is how you can search with Google Scholar also. So one thing is like you just have to search with any keyword of your interest based on your research area or based on your research requirements. Like say, I'm interested in bioinformatics tools for protein-protein interaction. So it will give me the list of articles which utilizes these things. So integrated web visualization for protein-protein interaction, algorithms and tools for protein-protein interaction. So based on your requirement, you can search, and then you can get literature, and from that literature you can find appropriate methodology or tools which are required for your research. So I'll quickly go through this Web of Science. I think Science Direct is straightforward, so I'll just show you maybe that one, Science Direct. So Science Direct lists you all the like list of journals, everything which is under the themes like physical sciences, engineering, chemical engineering, chemistry, all those things. Also life sciences, health sciences, and then social sciences and humanities. So if you click any of this alphabet, it will show you uh, journals which are under the title. So let's go for life sciences, and then biochemistry, genetics, and molecular biology. So here you can find journals like subdomain. I want biophysics, or I can go for cancer biology. So I'll go with biophysics. Here you can search journals, books, textbooks. So as I said, this is kind of publisher. Like Scientific lists all the publication under that one. So the journals can be linked to any publisher like Elsevier basically. So here you can find list of journals if you hit. So these are the journals available. 41 journals are available. So similarly, you can go through each of the journal and then read about the article. Like assume if you want to submit also, you can follow the same procedure to select journals. So to select journals also, there are tools which are available for you to select journals to submit your articles. So here you can read about the journal, what is the main focus of this journal. Like the focuses on protein structure conformation dynamics and protein folding, and what is the average time which they take to review and make a decision for the article submitted. So these are the given list. Same thing actually here. Okay. And this is the impact factor of your journal, which is calculated based on the citations in last 2 years, and you get list. Okay, so here also you can search, like say I'm interested in bacterial infection. I don't know it has or not, but let me just search. So bacterial pathogens. So they have published articles which are related to these topics, so I can read for my research. So individual journal wise also you can search. Otherwise, you can go collectively and search in the literature tools or databases which are available for you. And here to find journals also you can do one thing is like there are some things called as journal finders. So each like each of them have their own site. Like for Elsevier, it is Elsevier Journal Finder. So if you have a paper already, or if you have some known headings or keywords, you can just paste here, and then you will get it. Let me try one thing. Back. Okay, let's do one article. I'm just trying to paste the same thing. If you have abstract, you can paste. Otherwise, you can just paste a keyword also. It's fine. You have to give the keyword for your paper. So I don't know anything about this paper. I'm just giving stable wagon connector polymer. It is saying no keyword found, but maybe I should have taken a keyword from there, but it's fine. You can select area of research, field of research. I'm just giving based on the hierarchy, and then find journals. So based on my journal, it should list. So some chemical molecules which is like alginate polymer. So based on that, it will suggesting nano today, International Journal of Biological Macromolecules. So we got the same journal which it was published basically. So these are the journals which publishes similar articles. So you get the list of journals, also how much is the site score, impact factor, everything you get. Also you can use this filter to limit your searches, and also you can turn off here open access. If you don't want, you can keep subscription based or open access based. So basically you can find journals here also. So is there any questions, any doubts? If you have any doubts, any question, you can ask. Okay, if there are no questions, I'll switch to Web of Science, because Web of Science is little different. So this is from Clarivate Science Group. So here you can search journals to understand what is their metrics, like what is their indexing. There are two indexing, like SEA indexed, scientific collection index. So core collection is there, also core collection extension is there. Let's take the same journal and then just search. Only thing is you have to log in to your results here. So exact match found. So this is journal, and it gives you who is the publisher, what is the ISSN for the printed as well as the EISSN, and which comes under SCIE, basically indexing, scientific citation. There are two, one thing is SCI and then SCIE. expanded one. So these are the additional indexing they are. So if you click view profile, it may ask you to me to log in only basically. So, okay, so yeah, I'll just use Google login. So here you will get to know details about the journal. So journal website, when it was first published, what is the frequency they publish, like monthly, bi-monthly, annually, quarterly. You can get the list, how many issues they publish per year, and which country this journal belongs to, what is the language of publication, what are the index coverage they belong to, and which category. And you can also find similar journals based on this query. You can also search within this one. Here you can find, so here this gives you all the scores, which citation scores, indexing scores. You can also search within this same way. You can search for articles basically. So if I search for protein drug targets, it should give me a list of things which are present only specific to this journal. So I get so based on this, okay. Yeah. Actually, this is like more like subscription based one, Web of Science. So, but it will give you topics from there. You can also get articles basically. So here, what we searched is like we just search protein drug targets, and then from this particular journal, so it uses the search query like protein drug target as topic, and the journal. This is in here. It is instead of journal, it is called publication titles basically. So from here, you can get also the articles which are based on the query which you made. So like you can get articles, but I think recently only this channel has become open access, so you can get most of the articles freely available to from this journal. So this is one way to get articles, and other way is like Scopus. Scopus is basically based on institution access. So I'll not show that one because you have to have an institution access to search and make filter from Scopus. I'll show some of the secondary platforms which are available. So one of the other platform is called ResearchGate. So in ResearchGate also you can get articles. And also the advantage of using ResearchGate is like you can get the article information, everything. You can download the article directly from the ResearchGate platform. If say for example the author has uploaded their copy, or if the article is freely available like open access, ResearchGate automatically fetches from the site. Otherwise, one thing is like you can also request the article to be shared with you from the authors. So this article is there. So, but it doesn't have any full text available. So you can just click hit, and it'll just create a message like why you are interested in, and you can get a full text. And if the author sent you, then you will get a notification, and then you can read the article from the author directly. So directly you can get here. Also you can use the same search for a publication or for a journal or anything. You can search here also, like let's search bioinformatics drug discovery. So here there are multiple lists, like you can search publication, you can search researchers, projects which are listed under ResearchGate, or if some questions, also jobs, also institutions which work on those topics. So here publication based on what you want to, like article is there, preprint is there, conference paper, poster, even thesis and books are there. So I'm interested in only articles. It will give me the articles which are based on my query, bioinformatics and drug discovery. So I get articles here also. You can do similar search and then get articles for your literature search or survey. So other platform is Academia.edu, which is also similar one, but I will not recommend that. It is like you can, it's more of a paid one if you want to. Academia.edu actually. paid one to create your profile, but you can just search any articles. So it will give you, so basic search is like it will give you what are the hits available, and then so these are the full text article available and what are the titles of the article. You can use the same filter and all those things here also. What type of journals are publishing, even different languages, you can access all those things. So for this keyword, it has found about 700,000 papers, which is nearly impossible to read. So this requires a premium access to see what are the all results. You can just use basic search to just find article titles. If you find article titles, then you can just search or hit on it, and then you can get it from other sites, basically from PMC or from any other resource. Sometimes it gives you full text access here itself. You can download from here also. This is also one way to get. So other place where you can search recent researchers or new articles is bioRxiv. So this is a preprint server. So preprint servers are servers for research works which are not peer reviewed, basically. So every article which is published in journals are peer reviewed by scientific community, and based on the scientific community's suggestions, revisions and recommendations are made for these articles, and then finally it gets accepted for publication. So here in bioRxiv, so bioRxiv is similar to metaRxiv or another in terms of computer science and physics. So they use something called as archive. So this is inspired from that. So here also you can search for new articles or which are not peer reviewed, but something people are working and you can find. So there are like a ton of articles and topics available. So if you go to bioinformatics, you can just see what are the articles published, new result. So it will just list you abstract on the right side, but you can search if you have any specific ones. If you are looking at insulin, I'm looking at just search some times ago, I guess. So you can search. So based on that, you can get best match news first. So it gives me based on my query. So glutamine is an essential for overcoming immunosuppressing. So this has somewhere insulin mentioned. That's why it is listing me in the top. I think I saw an interesting result in the previous page. Insulin signaling and extended longevity in ants. So this is something new which we have never heard. So you can find new results and interesting results. Also, methodologies are being proposed here. You can look at those methodologies also. So similar way, you can search new articles also here. Like go to home. Here also advanced search option is available. So search term you want. What is the term you want? Like let's assume molecular dynamics. And then I am not limiting any search. So if I medRxiv, I don't think medRxiv will have. And subject of interest, I'll use bioinformatics since I used include articles from new results. So you can also publish and submit contradictory results or confirmatory results here. So authors, I don't have any preference. Title words, should I have any title word? This is a search term, but if I want to say, let's say algorithms. Abstract, this I don't want anything. So I'll just hit search. So basically I searched for an algorithm in molecular dynamics, which returned me 5,000 plus articles. And here you can find new articles which is published. So this is accessibility analysis and visualization for MD simulation data. So this is also MD simulation. They have used some algorithms. So based on the query which you give, you get the results like JX API for native Python interface for MD simulation with the plugin. So you can find article here also. Right side, if you go, you can limit your, filter basically your subject area. Like I want only the 2022 one. You can just click here from the shortcut, and it will give you the recent ones only. You can also sort similarly. with the same fashion which you did in the other databases. So it will give you mostly the results from all, since the query has been not taken now, because since I get hit only 2022, and you get some results which are unrelated to this also. So this is how you can access for literature search in bioarchive. So Scopus, preprint servers. Yeah, also there is another site which I don't remember exactly. So this is one of the biggest platform for searching queries basically. So I think F1000 research is also there. So you can get. So this is a like kind of publishing platform. It's like open peer-reviewed platform for research article. Here also you can find articles related to your interest or research. So here it is mostly I have to filter the queries, article type. I want research articles only. So research article. You can add multiple also, but it's fine. I'll just apply. So you can find research articles and which are like open peer review. And this is also kind of open publication. So you can sign version 1 peer review, awaiting for peer review. So similarly, you can find articles which are peer reviewed. So peer review 1 approved. We hit open and see. You can find the review status. What is the review report? What are the comments? Everything you can read about them. So the full text of article is freely available since it is open access. You can read it for free and then maybe adopt some methodology or from results which are useful for your study basically. So these are some of the literature search tools and platforms which are available for finding out articles, journals for your research interest. If you have any questions, you can ask now. I'll switch to the bioinformatics tools part in some time. So maybe we'll just pause here for 2 minutes. If you have any questions, any doubts, you can ask. Any questions? You can type in chat if you have any questions also. Any doubts, any questions? You can also directly unmute to ask. Okay, if there are no questions, I'll just switch to the next one. So like there are like a ton of resource for bioinformatics databases and tools which are available for your research. So mostly you use them based on your requirement. So it depends on what is your requirement. So based on your requirement, you can find tools and databases which are available. So the most comprehensive one which is available is NCBI. So in NCBI, you can find almost many databases which are listed. If I showed you like all databases, so you can see list of databases like assembly, biocollection, bioproject, books, CleanVar, this is TP, variation database, genome, gene, protein, peptide, mesh, PM, articles also, family, PubChem, PubMed, SNP data, SRA data, structure data. You can like browse through any of the data. If you want to see every data in every database, you can just search here directly. It will just give you a list of it's available in each database. So that I'm just searching cancer in NCBI and it gives me Cancer related things like 65,000 genes are listed, 39,000 bioprojects are there, biosamples are there, this many books, mesh terms, PubMed articles, PMC articles, domains. So you get the summary of your query here basically, and data sets which are available in GEO, Gene Expression Omnibus, and SRA, this is sequence read archive. So NGS data are available here. So you can just simply search here to retrieve any sequence or any uh, genome from for your research purpose. You can just choose your particular database and then hit the query keyword so you can get it. So let's assume I want a gene of insulin. I want protein. So I'm just typing Homo sapiens because insulin present in all other organisms also. So it gives me the gene here. I can also select from here. Why we get 2000 queries, 2000 hits is because it's based on submission from various people, and you can take the one which is listed as a reference one. So there are 4 reference proteins, 2 reference genes available. Since humans are eukaryotes, so they have splice variants or isoforms of the proteins. So you get a different length or slight variations in the protein sequence. So these are the protein sequences, and each have their own accession IDs. So this is called NCBI accession. If I open one, you can see the GP format. So, yeah, since it is protein, it's called GP GenPept. So here you get all the information: who are the authors, what are the organism, a list of information about this protein, and you can see the features of this one, like what is the length of the protein, details about the protein, molecular weight, where is the signal peptide located, what are the protein, mature protein length, so what is the CDS coding sequence, and what are the gene symbols which are there, and what are the other IDs which are available. So, and this is the basically the sequence. So if I want only the sequence, I can just go here, FASTA, and then it gives me the sequence. You can just copy paste, or you can just here file and then FASTA format, or any other format which is depending on your use case. You can just hit create file, it will download the file basically. Then you can use this sequence as a query for any other tools. So here I have listed almost many tools. So I'll just explain what are each of them. Maybe you can further explore what each one refers to. So NCBI is a collection of database from National Center for Biotechnology Information. UniProt is protein resource. So basically, if you are interested in looking at specific protein and then whole detail in one platform, you can go for UniProt. So this is the revamped new site for UniProt. So just go to, I'm just using the same query. I didn't mention the organism, so I'll just go directly human. It's list me human. And whichever protein is listed in gold color means reviewed basically. So there are 2 categories here: reviewed proteins and unreviewed proteins. Reviewed proteins are supported by evidences, also manually curated ones by the UniProt team. So here you get entry, feature viewer. So feature viewer just loads you the features of your proteins basically, like domain, transmembrane, what are the active site, like it will list you as domain only, but you can find those information. And what are the publications, other links, and what are the history or modifications in the database basically. So here you can, if you hit here, it will show you what are the domains present. It is showing fibronectin type 3 one domain. Here it is protein kinase domain. So you get a visual representation of your protein, and under that you get the structure of your protein. Basically, it will first list you PDB structures, like which are solved using different methods like X-ray, EM, or NMR. and also predicted structure, which is available from AlphaFold database. So if we hit AlphaFold here, it will open up a new site, and then you can get the same structure and information, how much is the confidence of prediction, all those information. And almost for most of the proteins, AlphaFold have predicted structure, and it is available in this particular database, AlphaFold protein structure database. Let's go back here. Here, if we click entry, okay. So here you can get the information. So the function is described here, catalytic activity, how it is regulated. This is the feature which we saw actually. So here it is given you as a text, but what we saw is like more of a visual representation, the collective visual representation. So here, these are shortcut keys. So if I click name taxonomy, it gives me the names, EC number for enzyme commission number, what are the short name, how it is cleaved into all the other details you get. What is the gene name, which organism it belong to, taxonomic classification, what are the accessions, primary, secondary, and from which proteome. Here in UniProt, you can also get proteomes, peptides, and from which chromosome it belong to, what are the other IDs available, and if you click here, what are the subcellular localization, like where this protein is expressed. Also, you get the gene ontology terms basically for the subcellular localization, like cellular localization, and which disease it is involved, and what are the variants, like what are the SNPs which are in the protein. You can look here, and what are the supporting evidences for these proteins, and what are the post-translational modifications which are known to in this protein. You can identify those also, and then the expression, how is the expression of this protein. You can get linked to different databases like Expression Atlas or Ensemble or HPA, Human Protein Atlas, and you can see the interaction, what this protein is interacting to. So here, I think it's not loading. Yeah. So here you can see what other protein it interacts to. So from here also you can look. Also there are specific databases. From there also you can look for the interactions available and structure which we saw there. So the same here also it will just show like what type of PDB structure, resolution, all the other details, how many chains, what are the residue. Direct link is also here. If you click here, it will just directly go to PDB, and you can get the family and then domain information. So insulin binding domain is there, disorder region is there, and then the sequence and isoforms. So as I said, like we have splice variants or mutations, so you get isoforms of the same protein. So isoform sequences are also provided here. And then similar proteins. So similar proteins will just give you what are the proteins which are identical to the query protein basically. So 90% identity you can search, so it will show you in different organisms. This is cattle or cow, and then I don't know about other organisms. So 50% identity you can just identify or get from here itself. If you are interested in doing some comparative proteomics or comparative genomics study, you can use this way to identify proteins from this one. And I'll just go back. If you have any questions, you can just ask directly. Any questions? Any doubts? If you have any problem which you are facing in this database or in finding any literature, you can ask. Okay, I'll just switch then. So this is a resource for protein sequence. So other one other than AlphaFold is PDB. PDB is structure database which gives you structures of macromolecules. So you can search here for any protein. Let's search insulin. So here also you get the code like structure. I go down, you get the PDB code here. So you can directly click here and open. It opens up to a page where it gives you the structure of your protein. Here also it lists you what is the paper which publishes this structure, what is the sequence length, how many chains are there, is there any mutations in this structure, and then the visual representation of the property of the protein, like what is hydrophobicity, what are the mutation disorder region, and then protein family which is present here. And then if there is any substrate information which is also given here, and ligands which is present during the crystallization process. So that information is also available here. And you can download the structure also from here. So you have PDB format or MMC format, or you can download the FASTA sequence also. So if you can click 3D view and then see the 3D view also. So let's go back to homepage. In homepage also you can search. So I search just the insulin, right? So it gives me a list of structures which is available in PDB. So I can limit to Homo sapiens only. So there are about 5,866 proteins available. I mean structures available. So if I hit this one, it will just load me those human proteins only. Okay, what happened? Yeah, it is showing only the human proteins now. So then I can limit X-ray structures. I want only proteins. You can choose the resolution, how much you want, and which year of publication, what type of enzyme classification, what type of symmetry, and then the structure classification of protein, that is scope, that also you can use. So I'll just hit here, it will just show me which is matching. So now I'm getting 437 structures only, but you should always look into each structure and then find, is it a complete structure or it is having any missing residues, or what only the domain is being crystallized, it's a monomer or dimer. So all this information you can go into each of the structure and then you can read about it. Also you can read more about, so here also they give you a link to PubMed. So if you open this one, it goes back to PubMed. If it is freely available, you can. Okay, what happened? I think I didn't click it. Oh, sorry, I think I clicked the wrong link. So here I have to click here. So it gives the PubMed literature for that article particularly. If it is freely available, full text will be accessible to you. Otherwise, you have to find it by requesting to the authors or writing to the authors directly to their email also. So there is a 3D view which shows you 3D representation of the structure. You can measure or do manipulations, like what you want to look into the structure, like look at particular residues or look at particular interactions with the ligand. All those things you can do here actually. So this is one way to look at the structures which when you select for your research purpose that you can continue. And there are like a lot of things here, but you can't look at everything, but you can just know the basics. There is also like to understand the PDB format, you can go here and then click this PDB 101, which gives you a detailed information about PDB, and then like what are the details available, how to look at the structure, and then what is the PDB format, every detail you can get basically from this PDB 101 site. So here you can look for paper model, 3D printing also you can do basically if you are interested in 3D printing, and you can learn about PDB here. And let me show you maybe like one resource for NGS data, which is like there are many resources like Geo, Array Express, SRA. So I will show you a different one which is called Omics DI. So basically this is a resource where you can search for omics related dataset, and it contains various omics like genomics, proteomics, transcriptomics, metagenomics, and then metabolomics, and it lists like multiomics also. If you are particularly interested in any organism, gene, or disease, you can search here. Maybe let's search for some disease. I'm not sure what disease I'll search. I'll just search maybe something like skin disease. So I just used a general generic query. If you have particular interest to any disease or any pathway or anything, you can just search basically. So here you can see at the left side, unknown is like not classified. Transcriptomics data, genomics data, multiomics, proteomics, metabolomics. I think models are from a big database, which is genome scale models, which are in SBML, systems biology markup language format. And pre-curated models are like curated by volunteers. Metagenomics data is also available. So if I go and hit transcriptomics, this is same as the same filter which we saw in every database basically. So it will just filter based on what. Okay, it is still loading. Yeah. So it will just basically filter you the data and it gives you the data. So here you can filter based on organism. So here this is what I mentioned. Like there are different repository like Array Express, GU, ENA. ENA is from EMBL, which is similar to SRA. This is European Nucleotide Archive. SRA is Sequence Read Archive. So Expression Atlas is also from EBA. So these repositories are combined here, and then this is more like a search engine or search portal for genomic or multiomic data available. You can also see psoriasis, melanoma, what are the diseases available, and then what tissue type also you can filter, and which instrument, like which platform which is used for sequencing these samples. And then if it is published, you can get the publication ID, publication year, and then ensemble ID, and what type. This we filtered already. And who are the filters? Like some institutions are regularly working on some particular area, you can filter with those also. So here it will just give you information, but again you have to get the data from the original repository. This is more like a search engine only. So gene expression profile of sensitive skin, if I click, and it gives me an abstract and details of it, and it shows me where it is available also. So it is available in GU and Array Express. If I click hit access data, it will go back to either of the resource, and then you can collect the data from here for your analysis basically. So it is more like a search engine for your omics data, where you can search for your data and then like maybe confine to this as the data sets which you will select for your study, and then proceed with it. So this way you can select and then search articles and then data from repositories, and other database are there, like string is one which gives us interaction information, string DB. So it gives you protein interaction information, and it contains almost many organisms. And the home page it was there, but you can also search. I'll just use the same keyword, insulin. So it is selecting and showing me, is this a protein which you are looking for? And then I'll just hit continue. So the red color is my query, insulin. And what are the other proteins it is interacting with? It is showing me the list of proteins it interact with. And there are settings you can modify to see what based on the evidence. Like each evidence is based on the color code which is representing here. So if you go to legend, and then you can see. So predicted interactions are these. Co-expression is black. Experiment is this. I don't know. This is a rose color or something like that. You can also filter here. So I want only experimental evidence. I can just remove all of these and then hit update. So I get experimental evidence for these interactions. So if you click here, you get to know what type of evidence is also there. So which protein is interacting with which one, and since these are disabled, so we are not getting this information. And similarly, you can also use a lot of other options present in analysis from here, which gives you gene ontology terms, publications related to your group of protein-protein interaction, cake pathways, reactome pathways, wiki pathway, and what are the disease associated to it, tissue expression, subcell localization, phenotype, everything you can get. As the other database for experimentally known protein-protein interaction data are intact, and the other one is BioGrid. I don't think I have mentioned here, but these two are the other resources which you can look. I'll just summarize these databases and then we'll just wind up here. So Stitch is for protein drug or protein compound interaction, which is developed by the same group by string database. HPADB is host pathogen interaction database. Between different pathogens and host, you can get the protein-protein interaction data like virus, plant or virus, human. And drug bank is drug information, which includes FDA approved drugs and other investigation drugs related to targets. Also, it contains PubChem is present in NCBA only, which includes publicly available chemical compounds and chemical informations. And TTD is therapeutic target database, which comprises mostly of human targets and other targets present in other eukaryotes and prokaryotes, including their drug information, and they also have their unique action ID. For pathways, you can use cake database, which includes not only pathways, but also you can get genes, genome, metabolite, drug also is available in cake database. PFAM is for protein families. You can identify, or for a given protein sequence, you can identify what protein family it has or what are the domain region it is present. Interpro is similar one, which integrates other platforms including Tiger, CDD, like conserved domain families. So it includes almost 10 platforms to predict protein domains, family, super family, all those information. And other tool, important tool, which frequently which we use is BLAST. So maybe you can I'll just show you BLAST. So I'll just take this question, this protein as a query. So there are like different types of BLAST. Nucleotide is for nucleotide query, and then protein is for protein query. And here it is like for translated nucleotide, like say protein query, and then you want to search protein. And then here it is like nucleotide query, and you can search against protein. In this, it is the reverse of that one. So we'll go for protein. And then I'll just submit, and then we'll come back to the original page. Yeah. So in this also we have different types of BLAST, like which database you want to search, RefSeq protein, model organism, or UniProt, or PDB. You can search, select structures, like say I'm interested in looking at structures of this protein sequence, so I can choose PDB, and then you can select which type of algorithm you want to use, like it's normal BLAST-P, or it should be position specific one, or it is like pattern. If you have specific pattern, you can give. So for now, we'll just use the normal BLAST, and then let it run. We'll just go here, and then I'll just look back what is left. So PHA is also pathogen host interaction database, and open targets is one platform where you get the targets associated with the disease with a score, association score for mentioned targets. So if you are interested in looking at targets for any disease or something, you can just go for open targets and then just search for any disease or any gene you can search basically. So go to this one, like if you have targets known, you can use that one. Otherwise, you can just search, like let me just click leukemia here. So it gives me associated targets are listed here, and these targets are ranked based on several categories like association, mutation, available drugs, pathways. Same way, you can also use the filters which are available for this search query. So I think the BLAST results have come. So this is our query protein, and against this database and length of the query, and then it gives me what are the structures available. So first one is like cryo-EM structure of insulin, and with an e-value of this much and with a identity percentage of 100. So basically this is the PDB ID which you can search, or you can just hit and it will go to NCBI page. Since this is search is based on NCBI, you can get. So based on the identity percentage, e-value and query coverage, one can select any protein. And you should also look about the structure details from this PDB ID. That would be more advantageous in selecting any structure for structure-based drug discovery or structure-based approaches for any case. So this is basically, and similar way you can limit your search to humans or any like 06 is the human tax ID. So you can also limit here like e-value how much you want. I want only 90 to 100% identity. You can list them here. E-value should be less actually. So let's query coverage should be 80 to 100. I'll give. I'm giving the highest one. So filter, if I apply, it will filter me those results and I'll give. Since I think I gave query coverage higher, maybe I'm not getting my 50. I'll give filter. Yeah. So these are the two query coverage, which is about 78 only, which we are getting. So if you click one, it will give you the alignment. So this is the alignment for your search query as well as the subject which is present in the database. And it gives you what is identity percentage and gaps, everything. These details you can get from this blast results. I'll just quickly go here and then tell about others. Like Cosmic is for cancer specific datasets. So one can identify or read about cancer genes based on publication and evidences, which is classified into oncogenes or tumor suppressor genes. So similarly for cancer, there is a database called TCGA, the Cancer Genome Atlas, which can also be used, and which has a specific platform called CBio portal, which shows you how genes are expressed, like your interest of genes are expressed at various publicly available TCGA data. And these three are databases for sequences, next generation sequences. And there are a lot of XPCAD tools like Schrodinger Discovery Studio. It can go on. And then there is open platform called as Use Galaxy, which you can use for running any tools without installation on web, which provides you 250 GB of storage to run your analysis, and so you can make workflows using Use Galaxy. I will show only maybe these two things lastly. XPC tools. So XPC tools covers almost most of the tools which is based on this SIV resources. So you can search based on individual category, basically systems biology, text mining, structural biology, proteomics, genomics. So if I click here, it just lists me protein based resources. So Swiss model, string database, which we saw earlier. This is UniProt classifier, MF tools, make2db, UniProt. So this is more like a database of databases, like it gives you list of database. You can find a lot of tools information from these XPC tools. And the last one I want to say is that Use Galaxy. Yeah. So there are different platforms available for Use Galaxy. So you can use Galaxy.org. So this is okay. I think there is a spelling mistake. Use Galaxy is the universal one. I don't know what it will load to. 1 second. So there is usegalaxy.org. There is usegalaxy.eu. Also there is a Australian server. Also there is a Indian server also. I think india.galaxy.eu. So this is a platform where you can learn. Also to learn Galaxy, there are free tutorials available online. Use Galaxy. Oops. something will open. Yeah, this is the platform for use directly org. There is no difference, but some tools are the only differences in Galaxy platform. One can learn freely about Galaxy in their portal itself under the Galaxy training website. So you can learn all these topics for free. With the tutorial, everything is available. Like, for example, transcriptomics, if I go, it gives me a tutorial and then hands-on, like single cell RNA-seq. I just hit this one. It gives me step-by-step instruction what to do, how to do in the Galaxy platform, what are the analysis types available. So everything can be learned from here for freely. I think I'll stop here. If there are any questions, you can ask. Any questions? Any doubts?